thanks again for being here. We appreciate it. Today we're very excited to have Dobi Ranev, also from our psychology department here at Georgia Tech, speaking to us today. Dobi did his undergraduate work at Harvard, did his graduate work at Columbia, ventured out to the West Coast for a postdoc before he came back to the East. It was 2015, is that right, that you started here at Georgia Tech? So assistant professor the last couple of years in psychology here at Georgia Tech. Combination of imaging, TMS, and computational modeling work. I'm very excited to hear what Dobi has to tell us today. So, Dobi, welcome.
Can everybody hear me okay? Yep. All right. Thank you so much for inviting me. It's a real pleasure and honor to present my lab's research to you. Today I want to talk to you about two main things. One is the organization of top-down control in perception. So this is work that I've been doing for a while, and it's more of a anatomical kind of work, so where in the brain do different things happen. And the second part is more recent work, and it's a little bit more theoretical, and it concerns the computations of in perceptual decision making. So let's just start with the first part. And by the way, if you have any questions, feel free to interrupt me and ask them. So you probably know the basics of how visual processing is organized in the brain, right? So information comes from the retina, goes to LGN, visual cortex, and then two pathways, ventral and dorsal. And I'm not going to go into the details, but what I want to highlight here is that when it comes to the frontal cortex, there's not much that we think is important. There's the FEF, the frontal eye fields, which control eye movements, so they're important. But besides that, the frontal cortex doesn't really do too much about vision, right? So what is the frontal cortex important for? Well, it's important for cognitive control. So things like switching between different tasks, deciding what task to do in the first place, response selection, and things like that. So very, very high level processes. And so this is from a particular paper that advocates that there's a rostral to caudal hierarchy, and there's a different paper, and there's a number of them that all advocate for this kind of a hierarchy where more rostral or more anterior regions are involved in processes that are more abstract, and the more caudal regions are involved in processes that are more specific, so less abstract. But in general, frontal cortex seems to be involved in these high-level cognitive processes, right? Now, perception, of course, is not just bottom-up. There's top-down modulations. So isn't it logical to think that the prefrontal cortex may be involved in that? So I actually think so, and I think that the normal picture of vision is in the back of the brain and cognition is in the front of the brain needs to be appended with how the frontal cortex is involved in modulating top-down perception. And today I'm going to try to convince you that there is a specific organization that one can discern in the frontal cortex, such that the frontal cortex controls three different classes of processes for perceptual decision making. So you're hopefully asking yourself, what are those three different classes of processes? So let me introduce them by giving you a task to complete. So I'm going to show you a picture, and I want you to find the blue fence in this picture. OK? Ready? All right. Not that difficult. Everybody found it? OK. So my question is, how did you actually do it? So if you're like most people, you probably looked in the middle of the picture. There was no blue fence there. Perhaps then you found the brown fence. It's a fence, but it's not blue. And then finally, you found the blue fence. The thing is, there's too much happening in this picture, so you couldn't really process everything at the same time. You need to select different regions of the picture sequentially. So you engage in a process of selection control. So you selected different parts of the image for further processing. A second thing to notice about what you did is your search was not random. So for example, probably very few of you spent any time looking at the blue sky, even though it features one very critical component of what you're searching, which is the color. It's the same color, it's blue. But you know how pictures are organized, and you can use that top-down information, this conceptual knowledge that you have, and you can combine it with the perceptual information you're getting from the image, and together you can use that information to guide your search. So this is a form of decision control that you're engaging in. Finally, once you found the blue fence, perhaps you ask yourself, well, is that really my target? Could there be a second blue fence? Is this a trick question? Maybe then you search a little bit more, satisfied yourself that there was no other blue fence, and decided that I know for sure that now that this is my target. So that was a process of evaluation. So am I really confident in what I think I'm seeing? And if I'm not confident enough, let me gather more information until I get more confident. All right. So these three processes are very broad. In order to study them scientifically, we need to operationalize them, right? So I'm going to operationalize them in this way. So for selection control, I'm going to study spatial attention. I think studying feature-based and object attention would give very similar results, but all of the studies I'll present concern spatial attention. For decision control, I'll focus on criterion setting. So what that means is I'm going to be giving subjects information about the stimulus before the stimulus arrives. And that information needs to be combined with the information from the stimulus. And if you try to model how this is done in the brain, it's really just a criterion shift. So that's why I call it criterion setting, because that's how we're going to actually measure it. And finally, for evaluation, I'm just going to ask people to give confidence ratings. All right, so let me tell you about a couple of studies that I've done related to each of these processes, and then I'll bring them all together. And I want to tell you about a recent study that we did that combines all three of them. So first, for selection control. So this is a topic that is near and dear to my heart. I can give you a separate talk on that. But I just want to highlight a couple of findings from my previous work, which is that, surprisingly perhaps, it turns out that what top-down attention does is induce a conservative bias in perception. So what does that actually mean? So what we did in this particular study is we had attended stimuli and unattended stimuli. And of course, people are better for the attended stimuli compared to the unattended stimuli. They have higher D prime, higher performance. But you can counteract that by making the unattended stimulus have higher contrast. So if you present the unattended stimulus with higher contrast, the attended stimulus with lower contrast, you can adjust the contrast such that D prime is actually the same. So their performance is actually the same for the two of them. Then you can ask, well, what happens to the detection criterion? So if it's a detection task, are people going to be more liberal for the attended targets or for the unattended targets? Again, they're the same performance, same D prime. So what we find in this kind of studies is we successfully get D prime to be the same, and then there's a pretty large difference in criterion. It doesn't look large here, but this really corresponds to about, so this is the Q. This corresponds to about 60% yeses. This corresponds to about 35% yeses. So it's really a 25% difference in the yes response. So what turns out to be the case is that for the attended targets, you're actually more conservative. So you tend to say, I didn't see it. it wasn't there. For the unattended one, you tend to be liberal. You tend to say, yes, I saw it even when it wasn't there. So we extended this kind of effect by doing the same thing. We matched D prime, but then we asked people to just give us a visibility rating. So which one looks more visible to you? And again, we get the same thing when D prime is matched, so that's very important. The unattended targets are judged with, in this case, 55% high visibility rating and only 30% for the unattended, for the attended. So perhaps very counterintuitively, attention leads to more conservative biases. So we replicated this kind of effect, but without queuing. And rather than queuing, what we did was we measured the spontaneous activity in the dorsal tension network. So if you don't know what the dorsal tension network is, it consists of bilateral FEF, frontal eye fields, and bilateral IPS. So there's a large swath of brain tissue in the IPS. So we took this network and we looked at the fluctuations over time. And then at random times, we gave people a task. And the way we decided what is high attention and what is low attention is by doing a median split on the activity in the dorsal attention network. So if the activity there was higher, then we call that high attention. If it was lower, we call that lower attention. And again, we replicated the same kind of conservative biases with higher attention. So for our purposes, what I want to highlight here is that it's the FEF, the very caudal part of the frontal cortex that seems to be involved in these attentional or selection processes. Of course, this is not anything particularly new. The dorsal attention network has been known for a long time, and it consists of FEF, IPS, and sometimes MT is considered part of it, sometimes not. And this kind of network has been demonstrated in animal studies as well. So for example, this particular one, they injected, or they stimulated FEF, and then they noticed that they recovered from V4, and the nature of the receptive fields had changed, showing that FEF does control the visual cortex in the back of the brain. All right, so this is all I want to say about selection control. Decision control, so again, to study decision control, you really need to have the perceptual information, and you need to give subjects some sort of non-perceptual information for them to combine those two pieces of information. So the way we did that in this particular study is we gave subjects a cue about what the upcoming stimulus is likely to be. Then after a short fixation, the stimulus appeared. The stimulus could be either expanding dots or contracting dots. And importantly, what we did in this task, at this point, subjects still didn't know how to respond. So they didn't know if expanding and contracting were mapped to index and middle finger or middle finger and index finger. And we gave them the cue only afterwards. So in this case, the index finger on the left coded for contracting motion, and the middle finger coded for expanding motion. So the cue could be two kinds. It could be predictive, in which case it was 75% valid. So 75% of the time, if I told you expanding, then the motion was really expanding. But 25% of the time, it was invalid. So I told you expanding, but it was actually contracting. So in some blocks, it was predictive, and in other blocks, it was non-predictive. So it was, we're going to call it neutral. So I'm going to show you a cue, but you know that it doesn't really predict the upcoming stimulus. So behaviorally, people are able to use the cues. So criterion beta here is the tendency to say expand in this case. And you may notice that in the neutral cue condition, people are very close to unbiased on average, which is what you would expect. And they're biased in the correct direction for contract and expand cues, but they're very far from being optimal. So this is what is optimal for the contract cue, so 0.33, and they're way above that. And this is what is optimal for the expand cue, 3. By the way, this is a logarithmic scale. So they can do the task, but they're very far from optimal. Now, what is more interesting for us is to actually look at the brain activation. So if we were to compare the predictive cues to the neutral cues, then what do we see in the brain? So what are those regions that are more active when you're trying to combine information versus when you're not trying to combine information? So it turns out that what activates is bilateral GOPFC on the right and on the left, as well as bilateral IPS again, intraparietal sulcus. And for each of these regions, we can also correlate to what extent, on a subject-by-subject basis, the criterion shift that you notice, so the difference between this bar and this bar for a particular subject, correlates with the fMRI activation for that particular subject. And you see that the correlations are positive for both GOPFC and IPS, giving additional support that these two regions are involved in decision control. So again, what is important for us is in the frontal cortex, it's DOPFC that seems to be important for this processing stage. Finally, for evaluation, there's many ways you can approach this question, but one study that I want to highlight is a study in which we asked whether the evaluation interacts between different tasks. So this is the stimulus that we gave subjects, so that's quite a bit non-standard stimulus. So it has 40 characters, and they're colored in 2 different colors. So for each stimulus like that, we asked 2 questions. First, were there more X's or O's? So you respond either more X's or more O's. And then you give a confidence rating for your response. And then are there more blue letters or more red letters? And those 2 things were completely decorrelated. And then again, how confident are you? So what we're trying to understand is, on a particular trial, if you're more likely to, if you're confident on one of the tasks, are you likely to be confident on the other task for that particular trial? So for each subject, we did a regression. And again, this is a trial-by-trial basis within a subject. And for this particular subject, we found that there is a positive correlation indeed. So if you're more confident on one task, then you're more confident on the other task. And again, the 2 tasks are completely separate. The difficulty of the 2 tasks doesn't correlate with each other. So this one subject, all of our subjects, all but one show a positive correlation, and there's this one that shows a slightly negative correlation. But overall, this is the Fisher transformed correlation R value. It's not a huge effect size, but it's a medium effect size, and it's very highly significant because it's so consistent within subjects. And one thing that we worried about is, well, is it that on some trials maybe you're just attending to the stimulus really well, and that's why you're confident for both tasks, but perhaps this is because you're just better at both tasks, right? So if you're correct on both tasks more often, then you'll be confident on both tasks more often, right? So what we did was we also looked at the correlation between the accuracy on the 2 tasks, and the accuracy actually didn't correlate. So it's not that on some trials you attend more and on other trials you don't attend. Your attention is spread out relatively uniformly, and it's really this specific effect on confidence that if you're confident on one task, then you're confident on the other task. So the way we conceptualize of this effect is that confidence sort of leaks between tasks. So if I have high confidence once, I'm sort of in a high confidence mode, so to speak, and we've done more sophisticated modeling on that, but I don't have time here to go into it. But the basic effect is that confidence leaks between different tasks. So again, we can look between subjects at the correlation between this confidence leak effect and metacognition. And metacognition I'm going to define as the extent to which confidence predicts accuracy, so how tightly linked confidence and accuracy are. And it turns out that this is a negative correlation, so the more confidence leak you have, which is, in a way a bad thing, right? You don't want to have confidence leak. You want independent tasks to have independent confidence. So the more confidence leak you have, the lower metacognition you have. And for our purposes, what is important here is we also looked at the gray matter volume in the brain and how it correlates with this confidence leak idea. And it turns out that the higher confidence leak you have, the less gray matter volume you have in this region in anterior prefrontal cortex. And this kind of effect has actually been found before as well. So this is our study. But there's at least 2 more previous studies that found that activity in the anterior prefrontal cortex correlates with your ability to evaluate stimuli. All right, so overall, what I want to highlight is that there appear to be 3 different top-down processes that are distinguishable between each other. And so the selection control seems to be particularly mediated by the frontal eye fields located in the caudal prefrontal cortex. Decision control mediated by DLPFC in the mid prefrontal cortex. And evaluation, which is mediated by anterior prefrontal cortex, located in the rostral prefrontal cortex. Any questions at this point? All right. So one question that you may want to ask me is, well, OK, so all of these studies are correlational. And you obviously picking and choosing studies, and you're painting a hopefully somewhat convincing picture. But really, is there a better evidence for this kind of organization? So what we thought that would constitute a really good evidence is if we have causal evidence for this kind of organization. So we used, so how do we test this? So we used transcranial magnetic stimulation to target each of these 3 regions and see if we can disrupt just the corresponding stage of top-down control. So because we need a control site, we used the somatosensory cortex, S1, as our control site. So this was a long study. It was a 6-day study. So on day 1, we trained subjects with the task. It was a complex task. I'll show you a little later. On day 2, we scanned people with this task. And we used it to identify the regions of interest. So we used it to identify where these regions were. So for FEF, you can see that, so this is the location of FEF for different subjects. This is the location of DOPST for different subjects. For S1, we just use anatomical landmarks in the post-central gyrus. And APFC, we didn't get in our fMRI contrast, so we just use an anatomical coordinate. So it was the same location for every single subject. You may see that some of these dots here get pretty close to APFC, so I'll go back to that a little later. So the important thing happened then on days 3 to 6, in which we gave TMS to S1, FEF, DOPFC, and APFC in counterbalanced order. So the order was different for different subjects. So how did we manipulate the three processes, right? We have three processes that we want to target. So the first one is selection control. In order to tap that, we showed subject spatial cues, so we tell them attend to the right, not to the left stimulus. For decision control, we couldn't use the previous manipulation that we used because it was getting too complex. So we did a different manipulation, which was to ask subjects to be either fast or accurate on every trial. So in a particular trial, they may be asked to be fast. So again, this is a non-perceptual information that you need to incorporate into your perceptual decision making. So you need to adjust your decision making so that it's either fast or accurate, right? And finally, for evaluation, we just ask for confidence ratings. So this is what the task actually looked like. We gave them the two instructions, to the spatial cue and the fast versus accurate for 1 second. Then the stimuli appeared for 200 milliseconds. Then they were post-cued to respond to one of the two stimuli. So the post-cue was valid 75% of the time, invalid 25% of the time. And for the post-cue stimulus, they needed to say if it's a right-tilted or left-tilted, that is clockwise or counterclockwise orientation. And afterwards, they gave a confidence rating. So in this particular trial, the focus is to be fast and the cueing is valid. But obviously, on different trials, the focus was to be accurate and cueing was invalid. And all of this was counterbalanced and interleaved with each other. All right, so let's go to the results. So our measure of selection control was the reaction time for invalid trials minus the reaction time for valid trials. So what that means is if you're attending where I'm asking you to be attending, then you'll be faster for that location. When I ask you about the other stimulus, you'll be slower, right? So if you're following my attentional cue, then this is going to be a positive quantity. And if I disrupt your ability to attend, then that quantity should decrease. If I completely obliterate it, it should go to 0, right? And the prediction here is that TMS to FEF will affect the selection control. So here's the results. So this is after S1. Again, this is the control that's supposed to be doing nothing. So this is our baseline. So there is a strong positive quantity here. People are attending where we ask them to attend. And after FEF, this decreases, so people are not able to attend as well as they were able to attend after TMS to S1. For decision control, we have a similar measure, is the reaction time for accuracy focus minus reaction time for speed focus. So again, if you're following my instructions, then you'll be slower for accuracy, you'll be faster for speed, so this is going to be a positive value. If I disrupt your ability to follow the instructions, then this quantity should decrease. The prediction here is that TMS to DOPFC will affect decision control. And again, this is the baseline. And after DOPFC TMS, we get, again, a decrease in this value. So TMS to DOPFC did decrease your ability to exert decision control. Finally, for evaluation, we're just going to use metacognitive score here. So this is just how tightly accuracy and confidence are related to each other. And the prediction is that TMS to APFC will affect evaluation. So this is, again, exactly what we found. However, there's the weird thing that the effect is actually in the opposite direction. We we're expecting that the APFC would again decrease your ability to perform evaluation. In fact, it increased it. We're still trying to understand exactly what happened, and we're doing some follow-up studies on that. But what is important is it had an effect, right? It changed from baseline, so it does seem that that patch of cortex is important for that process. So I've only showed you, yes, Bilal.
Did you tell us what the protocol for TMS was? Is it during the whole trial, or is it just during?
I didn't. I should have. So the TMS protocol that we used was an offline protocol. So it's called data burst stimulation. You give it for about 40 seconds, and those 40 seconds you give about 600 pulses. And then the tissue below is inhibited for 45 minutes to an hour. So we first gave TMS, and then during that 45 to 50 minute period afterwards, we gave them the task. So I've only showed you so far the results that we sort of predicted, but let me show you the full results. And the full results show that the effects were pretty selective. So when you looked at selection control, whether you give TMS to S1, DOPFC, or APFC, there's no significant difference, and it's only TMS to FEF that leads to a decrease. For decision control, TMS to S1, FEF, and APFC doesn't make a difference, and it's TMS to DOPFC that decrease decision control. And for evaluation, again, this is the baseline, and we do get a decrease for both DOPFC and APFC, so this is the only place where the effect is not fully selective. But I want you to remind you that so for a lot of our subjects, DOPFC was actually localized in a very anterior position. So we think that explains those findings. In fact, when we do a median split on these subjects, and when we look at just the more anterior ones, they look more like APFC, and the more posterior ones look more like FEF. The effect was not significant, so I don't want to make too much of it. But it does seem that, overall we do get this progression where more rostral regions of the frontal cortex are important for later stages of the top-down control. So we didn't actually think about that in advance when we were designing the study, but later on we realized that, well, these three processes are pretty sequential. So in general, you exert selection control first because you first need to select the stimulus. You exert decision control only after you've selected the stimulus, and you evaluate only after you've made the decision. Now, some of these may overlap to some extent, so I'm not saying it's fully sequential, but there is, in general, a progression to these three processes. So we can actually analyze the activity in the instruction period. So the first 1 second before the stimuli were actually presented. In the decision period, which we defined as the stimulus from the beginning of the stimulus until they give the response. So this is the decision epoch. And the confidence epoch, which is since the time they gave the response until the time they gave their confidence response. So we can analyze the brain activity in those three epochs to see, are we seeing a progression towards more rostral regions as well? So that's exactly what we are finding. So this is the activity that we get for each of the three epochs. And probably there's a lot going on in the figure, but focus your attention on the frontal cortex on the left side, perhaps. So in the instruction, you only get very caudal activation in FEF. In the decision, this activation expands, and it moves also more anterior. And for confidence, it moves even more anterior. And we can combine all three of these in the same picture. And you see, and by the way, those dots are where we happen to stimulate. So the stimulation sites were chosen completely independently from this analysis. so you can see that you have this progression in the activation. And for these three ROIs, you can actually plot the activity for each epoch. And you see that for FEF, you have more activity in the early epochs compared to the later ones. In DOPFC, you get this inverted U shape, so most activity in the decision period. And for APFC, you have the most activity for the later epochs. So overall, this analysis sort of paints a very similar picture, which is that the later processes related to perceptual decision making are mediated by rostral regions of the frontal cortex. Now, one problem with this analysis was that the regressors were actually pretty correlated with each other. So the bolt activity is very slow, and if you have periods that are right next to each other and there's no jittering, then you get some correlations. So we think that the ground truth may be a lot more selective than what you're seeing here, and we're actually doing this study right now where we're introducing more jitter in order to see perhaps more selective results. All right, so just to summarize this part, I hope to have convinced you that there are three different top-down processes in perception, namely selection control, decision control, and evaluation, that later stages of this temporal hierarchy are mediated by progressively rostral parts of the frontal cortex. And I've also argued that this organization may be based on the intrinsic prefrontal structure. And again, this is actually a very similar organization to what has been argued to exist in cognitive control, where more rostral parts of the frontal cortex are involved in more abstract stages of cognitive control. Do you have any questions so far before I switch gears? Oh, yes?
I just you were surprised at the frontal cortex stimulation. Does that jive with the idea that it's mostly for inhibiting activity rather than?
There are many possibilities. It could be something like that. But what I do think is a little bit more likely is that, so we have a TMS protocol. We think that it always inhibits, but it may be that it inhibits only certain brain areas, certain neuropopulations, certain psychoarchitectonic regions, and for others, maybe there's a different effect. It's a little bit up in the air at this point. All right, so I've talked a lot about the anatomic organization of top-down control, but I'm also very interested in the computations, right? What about the computations? I've told you a few things here and there, but I haven't talked about the computations too much. So in the second part, and I'm going to be very brief here, I want to tell you a little bit about this. So computations, perceptual decision making, I'll introduce a couple of popular models, and I'll talk about the concept of uncertainty representation, and then mention the implications. All right, so this is going to be a busy slide. I'll introduce 4 different theories of perceptual decision making. So hopefully you've heard about most of these. So the first one is the drift diffusion model. The idea here is that if you're making a decision between 2 different options, then you have this particle that diffuses over time, and it reaches one of the boundaries. And when it reaches one of the boundaries, you give the decision for that choice, not for the other. And how long it takes to reach the boundary determines the reaction time that you have. Signal detection theory is a much older and simpler model where each choice gives rise to a normal distribution over many trials, and you give your choice based on a criterion or a threshold that you can set either in the middle or you can set it elsewhere. Probabilistic population codes, in this kind of a scheme, you have neurons that code for different orientations. So for example, in a motion task, you can have neurons that code for different orientations, and orientations that are far away from the actual motion give low activation, but the ones that are right where the motion direction is produce higher activation. And then you can, this population activity, you can pass through a Bayesian decoder and you can get the full distribution of activity. Finally, deep convolutional networks. This is not formally really a model of perceptual decision making. It hasn't been used this way so far, as far as I know, but it's a very useful model to think about, and it's getting more and more popular. So it consists of multiple layers that process the image until in the final layer you have the output, and you can just read the output from that final layer. So these models are different in many ways. One way is that drift diffusion only applies to two choice decisions, really. Same for signal detection theory. Probabilistic population codes apply to continuous quantity decisions, and deep convolutional networks mostly apply to n choice decisions. So n can be a lot higher than 2. But what I want to really emphasize here is what kind of information you get from each model. So the only thing you get from the drift diffusion model is which boundary you crossed. So in a particular trial, you get that one point, and this is the information you have. So this is the choice, and perhaps you can add the reaction time. So you know also what is the reaction time. For signal detection theory, on a single trial, you get a point here. So you get the choice again, is it on the right or on the left of the criterion? But you also get a strength of evidence. So how far is this point from the criterion? For continuous quantity decisions, you get the whole distribution. And for n-choice decisions, it's not so clear. Again, they haven't been used in this kind of context. But you can argue that, again, it's choice plus strength of evidence, just like in signal detection theory. Or you can argue that, well, you can get a distribution across all of these. But again, it's not so clear there. So this idea of alternative representation, let me distinguish two different possibilities, which is cognitive uncertainty and sensory uncertainty. So if we imagine that we are better, and there's this pitcher who threw a pitch at us, and this is the strike zone. Cognitively, you may know that this particular pitcher has 3 main pitches: fastball, curveball, and changeup. And those 3 pitches tend to land in these locations with certain distributions. So this is just, before you have even seen the pitch, you know that information. So this is the cognitive uncertainty. Now, the sensory uncertainty would be what you actually get on this particular pitch, right? So you see what is coming at you. And one extreme possibility is that there's no uncertainty in your representation. You just think that the pitch is going to land right there. So you have just this one spot and nothing else. Versus another extreme possibility is that you have the full distribution. So you know the center of the distribution, but you also know the exact shape of the distribution. So just to make, so these are 2 very extreme cases, but it's actually more possibilities in between. So let me introduce those to you, and this is really my take on this. This is not necessarily something that is accepted in the literature. But if you have a stimulus, which is a moving bar, and you want to estimate the direction of motion, and you have this underlying distribution over many neurons, Then scheme 1 would be you have no uncertainty, so you just represent the most likely orientation, and that's it, nothing else. Scheme 2 is you have this indirect uncertainty where you represent the most likely orientation, but you also have some additional things like reaction time or how much I attended on that trial that you can use to gauge whether or not your estimate is good or not. Scheme 3 is, again, you have your estimate, but you have this strength of evidence. So if that strength of evidence is high, then you can be pretty certain. If it's low, you're less certain. Scheme 4, now you have a distribution, and for that distribution, you have the mean and you have the standard deviation, but you don't have the whole distribution. And scheme 5 is you actually have the whole distribution, right? So you get, as you progress here, you get more and more explicit and complete uncertainty representation. So diffusion. So how does each of these models fall into these schemes? So diffusion really has representation of scheme 1 or 2. Signal detection, because you have the strength of evidence, falls into scheme 3. This is a full distribution here, so scheme 5, and not so clear about the deep convolutional networks. So I'm running out of time, so I'm not going to go into all of the details here. But I want to say that what we did to distinguish between these things is we looked at pre-queuing versus post-queuing. So if you give a queue for the subject before the stimulus arrives versus after the stimulus arrives, then you can actually distinguish between these different schemes. So let me show you the results of this. So when we compare the sensitivity for the pre-queue when there's a predictive queue versus the neutral queue, you get no difference, and the same for a post-queue. So your ability to process the stimulus doesn't change based on the pre-queue or the post-queue. But this is how your criterion changes for the pre-queue. So you're able to use the queue. And weirdly enough, for a post-queue, you actually get even higher difference in the criterion. So the post-queue is actually more effective at changing your decision than is the pre-queue. So what that means for our models is the drift diffusion really cannot account for that, because it doesn't even know how to deal with a post-queue. Because once you have the choice, a post-queue is pretty much impossible to be incorporated. The signal detection theory, maybe you can account for this kind of effect with the recency effect. So the more recent stimulus has a bigger effect. But it's hard to think how you're going to account for this kind of effect if you have the whole distribution. And again, who knows for the deep convolutional networks. And there's some other ways in which you can distinguish between the 5 schemes. They pretty much give the same idea that it's scheme 3 that looks the most likely. So having the estimate of the stimulus plus a strength of evidence seems to be the most likely representation. So what I think the field should be moving in, and what I'm trying to do, is moving away from the standard representation of decision making, which is there's a stimulus, there's sensory processing, there's decision, and there's motor response, and all we care about is the decision. So that's really all of the theories. All they care about is the decision, and we don't really say much about the sensory processing or the motor processing. And I think it's actually really important to look into this, to look into that, in order to understand how reaction times are, arise from all 3 of these stages. And that's why the deep convolutional networks, I think, are very promising because they're very explicit about this stage, unlike all of the other theories. No theory is explicit about this stage. We should add that as well. The other question is, where is the decision stage? If you know Michael Shadlin's work, he thinks it's in LIP. There's a lot of recent work that says it's definitely not in LIP. So the question is, where is it? And there's also a second question of, do we really know that that stage exists as a separate stage in its own right? And finally, it's very important to realize that all of our optimality claims, and there's a lot in the literature, are based on a particular model that just takes the sensory processing for granted and then looks at the optimality of the decision stage. But if that model is not completely true, then all of those claims just fall apart. And I think a lot of our claims about optimality are actually misguided. So I want to thank my lab and my collaborators, and thank you for your attention. I don't know if you have any time for questions.
Maybe one brief one. I have a question. I have a brief question. So you mentioned probabilistic population codes, which would be a population representation of the entire distribution. The competing hypothesis to this is that neural systems represent distributions by actually sampling the distributions rather than representing them explicitly. Do you have any sense of how you could distinguish that sort of hypothesis from the others?
Yeah, so that one is tricky. I actually have thought about sampling a lot, and that's a hypothesis that's a little bit less emphasized in the literature in terms of connecting it back to behavioral results. The sampling is weird because if you sample a lot, then you get a full distribution, and everything that I said about probabilistic population codes apply. But then if you only have 3-4 samples, then perhaps that becomes more similar to signal detection theory. So because it's a very flexible theory in some way, I'm having a harder time pinning it down and bringing behavioral evidence to either confirm or disconfirm it.
All right. Well, let's thank our speaker again.